I wanted to thank Melissa and Fraser for inviting me today to present something a little bit different. And of course, thanks to the Foresight team. It's always a pleasure to see you and to attend these wonderful workshops. So I want to come back also to one idea that we had earlier. A tool that we need is to measure force at nanoscale. And in fact, this is what I'm going to talk to you about today, probing individual molecular machine using single molecule techniques. So really to probe one molecule at a time. So I will show you briefly some techniques that we can use. And today I didn't want to show my result, but show mainly what this technique can bring you if you want to collaborate one day with us or some other group maybe. So the first idea is the atomic force microscopy. So it's the first technique that I want to talk to you about. So this is a cantilever with a microscopic probe, and we can try to catch a molecule between the tip and the substrate, and you can detect forces of the range of piconewton. Probably most of you know the AFM for its imaging technique. So here is an example. You have different modes, of course, of imaging. And recently, there's been shown that you can use an AFM tip to really get a nice image of single molecules. You also have nano manipulation. But today, I want to talk about another thing, which is single molecule force spectroscopy. So really probing one molecule and exert a force on this molecule. So this is the kind of experiment that we are doing in our group in Liège. So we are catching one molecule at a time, and we exert a force on this molecule, and we can get the force resistance of this molecule, and sometimes we can have information about the conformation. If this is a molecular machine, we can have information about the processes that are involved in the behavior of this molecule. And we get that from what we call the force curve. So it's the force that the molecule is exerting against the pulling direction versus the tip surface distance. So the first work that was performed in single molecule force spectroscopy using AFM was in 1998 by Hermann Gaube in Munich. So it was, he studied the muscle protein titin, which was stretched by AFM. And in fact, so the titin is made of different IG domains. And when you stretch the molecule, you can unfold these different domains one after the other, which lead to this series of peaks. And if you have a look in more precision into this peak, you can see that the distance between this peak corresponds to the opening of each IG domain. So this was really the first work on proteins. Another technique that I want to talk to you about is tweezers. So we have optical tweezers and we have magnetic tweezers. And the idea behind this is that you can trap a bead in a laser, and one or two beads, and you can have one molecule that is attached between these two beads, and you can stretch it. So it's the same principles you stretch a molecule, except that the optical tweezers have another resolution, so you can go to really low forces, and for AFM you don't have that. But the drawback is that you cannot go to really high forces. So the two methods are really complementary. So first I want to show you that we can probe biological molecules. So we have a lot of examples of proteins that were stretched by AFM. You can have antigen-antibody interaction that you can characterize in terms of force. You can have also the study of the conformation of polysaccharides. In the lab, we studied the behavior of DNA under mechanical stress, but there are a lot of different studies on DNA. And the last work that we will submit soon is the study of polypeptides. So we know that these polypeptides make alpha helices in solution. And there's one information that is missing in the literature, is that when you have an alpha helix, you cannot have this alpha helix intact over a really long distance. So you have a critical length in terms of number of residue to have an intact helix. And in the literature, you can see that there are only simulations showing that above 20 residue, you don't have an intact alpha helix, but you have kinks in this alpha helix. And here, this is the first time that we show, so it's with polypeptides of 90 residues, we show that we have different kinks, and we can see that by the plateau here, which has different lengths. So we have, sometimes we probe 1, 2, 3, or 4 helices of 20 amino acids. So these were some examples in biological molecules. We have also biological machines, and here the example that I will show you were studied by Optical Tweezers. So they studied the unzipping of nucleosomes, so in 2017. Really recently, what they studied was the ATP synthase. So it was reported, so they could trap really a bead that was attached to the ATP synthase, and they could really follow the movement of the ATP synthase using the bead, and you can see that you have an increase. So this is the number of revolution of the bead using the ATP synthase. So this is really, really new, and it's I think really nice in the field. You can also follow the movement of myosin on actin using this microbead, and also recently it was show that you can follow the kinesin motors on a microtubule. So you can really have a lot of information on biological molecular machines. And what we wanted to do was to go to artificial machines, artificial molecular machines. So why do we want to do that? Because we want to compare the two, natural and artificial systems, to see if we are able to compete or surpass the performance of these biological machines. Using the single molecule technique, you can have access to singular behavior. So if you use ensemble technique, you will have an average behavior of all your molecules, thousands of molecules. Here, using single molecule technique, you can have different singular behavior depending on which molecule you are probing, which is really important, and you can have different kind of information depending on the molecule you are studying, so the actuation force, the reversibility, and so on. So of course, it's mainly fundamental research that we have now. So we need to increase our understanding of molecular interaction, the conformation, the processes of molecular machines. You have also a lot of instrumentation development, because all the molecular machine, especially the artificial one, are not probed in water or in buffer. So we need to increase, to change the solvent, so we need to change also the instrument. And I wanted to quote also something said by Ben Feringa, saying that nature leads the way when it comes to motors on the molecular scale, but chemists are in hot pursuit. So we are really following the nature, even if we use different routes. And we design controllable structure that can mimic muscles or rotary motors. So of course, we probed some MIMs, mechanically interact molecules. And I really believe that in order to design atomically precise molecule, we need to understand the behavior and their working processes. And if it's not by ensemble technique, it will be by single molecule techniques to really have a precise description of the MIM at the nanoscale before moving to some applications. So in this field, there were some examples by our group. So the first one was in 2011. So it was the probing of Rotaxan in collaboration with the group of David Lee. So we stretched a ring. So this ring was attached to a peg, and we attached the peg to the AFM tip, and then we can stretch the ring here to pull it to its unpreferred station. And when we release the tension, the ring is able to exert a force against the pulling direction to come back to its preferred station. So we can really detect the work performed by the ring to come back to its preferred station. There was also some work on catenanes at the single molecule level, showing that depending on the conformation of the catenane, you can have a different flexibility or rigidity in your chain. And also last year, there was this study published by Borja Ibarra in Madrid. And in that case, they use optical tweezers. It was a system that was close to this one, except that, as I said earlier, they could go to a really low force. So with optical tweezers, you can say, okay, I will apply a force of 4.5 piconewton, for example. And by changing the force they apply, they can change the potential energy of the system, and they can change the fact that the ring wants to stay on one station or another. So the work that I performed during my thesis in collaboration with Fraser was the study of this oligorotaxane. And in fact, so it was, we wanted to probe this molecule to see if the folded conformation was still there in solution and under tension. So we apply a force on this molecule, and then we release the tension, and we see that the molecule wants to stay in this conformation, so exert a force to come back to this folded state. So we compare this with different studies on proteins, on folding protein, and we show that this synthetic system was able to surpass the performance of folding proteins. What I'm working on right now, it's probing another kind of molecule, which is the molecular knot, again with David Lee. And we show that we can tighten a knot, and we can see what is happening when we exert a force on a molecular knot. So we have a sliding of the chain inside the knot, and we can detect that by AFM. So this was an overview of what we can do in the lab, so on rotaxane, catenan, knots. We are also probing rotors. I did not talk about this today, but we are probing also rotors in the lab. We are also focused on mechanochemistry, so we are trying to understand if some reaction can be reversible under an external force. We are probing helical structures, either bio-inspired or more rigid structures that are synthesized, and we are also probing DNA. Of course, we are relying on different collaboration. So if you want to add one part to this panel of research, you're welcome to come to me, and also to contact my PI, Anne-Sophie Duet. So some take-home message concerning this single molecule techniques to study artificial molecular machine. So these techniques are now powerful tools to study the conformation and the processes of this molecular machine. You can have access to fundamental understanding of the working processes at the single molecule level, and you can detect different behavior of the same molecule. Now that the AFM and optical studies on MIMs are known, so we have the first example, and now we can also go towards the thermodynamic and kinetic description of those systems. And to come back also to read the theme of this workshop, I wanted to say that we really need now a synergy between the organic chemists, material scientists, engineers, computer science guys, to really for the molecular machine to be a part of contemporary material science. So I think this is really the idea today, to gather all these people, to get really new ideas and to have new collaboration. So with that, I would like to thank, of course, the team, our collaborators, and I'll be happy to answer a few questions. Thank you.
Thank you. Plenty of time for questions, so fire away.
Damien, how do you decouple the flexibility of the linker from force measurements?
So if I may take an example, if I come back to, let's say, this one. So when you stretch this molecule, so you want to really see the movement of the ring along the thread. First, of course, you pull on the linker, and you have a specific behavior of the linker at the beginning of your curve, and then something happens. If you only have a linker, if you pull on a peg, you will have only a single peak. In that case, for example, you will have the beginning of the peak, and then you will have the force that will decrease for some point and then increase again. So you have something different in your behavior. So you need to, of course, decouple it and be sure that it's not only your linker. But
Linker can have several transitions depending on the chemistry.
yeah, it depends in which solvent you are doing the experiment. But we always have a blank using only the linker alone. And usually we use the PEG linker. And in all the solvent that we are using, it's mainly a single peak. And we can detect that. So we have statistical analytical tools to fit this, the behavior of this PEG and compare with the force curve that we have for the molecule.
Very interesting. If you cycle the unfolding process of a tethered protein, do you get the same force distance curve? In other words, does it refold the same way when you relax the stress?
It depends, really. In terms of proteins, I've never worked myself on proteins. I can see a lot of papers. Usually, for example, for the titin, I know that the reversibility was complete, so you have exactly the same. But of course, for more complex protein, you have misfolding, and so you don't always have the same over several turns. And we are also doing what we call the pulling relaxing cycles, so we can do this several times. And we showed in our paper that sometimes you lose some energy, sometimes you gain some energy, and the process is always different, and you try to find how the molecule behaves most of the time.
Hi, Damien.
Yeah.
Very nice talk. So my question is, you show one slide about like optical tweezer, EFM, and also magnetic tweezer. So can you, like in terms of like row taxing or catenating structure, what's the advantage or disadvantage when you apply different kind of technologies on the same type of the molecule?
I mean, it really depends what kind of information, what kind of interaction you want to probe. If you want to go to mechanochemistry, so really to go to covalent interaction, then you need to use AFM, because you need to go to higher force. If you are probing really small changes in the conformation, you can go to optical tweezers. So it depends what kind of interaction you are probing.
I was just going to make a small point in response to Joe's question. I know of other people stretching and relaxing proteins, millions of times and not generally seeing misfolding, but when they placed in the laser tweezers suspect prion disease proteins, they do now indeed see the misfolding events with large statistics. So it's become a tool for prion disease study.
Thanks, Damien. I'm wondering, like, have you think about like to take this one to confirm the protein, the conformation of the protein? I mean, generally now, it's like people use the AMR and crystallography and cryo-EM to do that. So I'm wondering, like, it's possible, just like, for example, this small protein, and we just want to determine the conformation. Is it possible just like functionize the tip with a certain function group over there, and we just touch the protein, and then to do that, probably we're going to do, it's like thousands of, stretching, and then finally we know, like, which, fragment is on the surface, and then generally combined with the, the information from the AMR or some other information, then we know the entire conformation or folding structure.
I'm not sure you can use that as a unique technique to get the conformation, because you get only the force curve information, and from that you have to, it's like a puzzle, you need to figure that out, to figure what's going on in your system. So I will not say to someone, okay, just give me the molecule, I will give you the conformation. That's not possible, and that's nonsense, because we have a good instrument to do that. We are not trying to replace that, it's just that we have different information with that, with single molecule techniques. So I think if you want really to get the conformation, you have a lot of ensemble tools, but then if you want to confirm, so in the case of the oligorotaxane that we studied, so this one, we could confirm by the opening of so the breaking of intramolecular interaction, we could confirm that yes, it was folded. So we have the information from in solution, but we wanted to confirm it by saying, okay, we break the interaction and they are recreating. We can confirm that. So it's the basis, but I cannot say if you give me a protein, I cannot say it will be folded that way.